Just to start off, we are a Danaher company. You might see a lot of familiar names here as far as our sister operating companies. Broadly, we really are focused on delivering solutions in terms of science and therapeutics development. At Cytiva, again, we are not developing therapeutics. Our goal is to provide solutions for our customers to leverage in their journey in developing therapeutics. And in the current context of this presentation, I'd like to cover really a short story focusing more on targeted LNPs for this applications. And if you think of targeted LNPs in this kind of in vivo CAR T application, they're, again, quite a complex biological molecule, if you may. What really matters in terms of how to use them is honestly controlling their composition, what cells they work in, as well as kind of the surface of the molecule itself to ensure kind of distribution in the right cell types. If you think of it kind of from the end, you want these LNPs to be able to be delivered and express the RNA that they encapsulate in specific types of cell, which means taking one step back, you want them to be close to that type of cell to be uptaken into them. So controlling by distribution of these kind of LNPs is important. And again, taking it one step back, that means from a design chemistry perspective, you're looking for a particle that has the capability of, again, engage with the right protein corona to be in the right environment, really, to have the chance for all this to come together. For this study specifically, we're using several platforms that Cytiva has, but really in the context of looking at this LNP drug product, target LNP drug product from a holistic perspective, in terms of, again, route of administration all the way, tracking the molecule all the way to the cell of interest, if you may. The platforms we are leveraging, just a quick highlight here, we're leveraging here the chemistry platform we have here at Cytiva on both the cationic ionizable lipids that we've developed and really read it for a variety of different biological applications, but also stabilizers, if you may. These are molecules that are usually on the surface of the LNPs, and they modulate the surface chemistry of LNPs, and therefore also can modulate what kind of protein coronas are engaged in the body, also can be used to attach moieties to the surface of LNPs. Beyond that, we leverage also our nanoassembler manufacturing platform, which really is focused on, again, providing a manufacturing process that is, in a way, the same at bench scale as it is in a clinical commercial setting. And then we achieved that with our next-gen hexing technology here. And why this is important is honestly control over the size of your particles, because not only the physicochemical properties of your particles matter, the size of the particle will have a direct impact on biodistribution in the body. So manufacturing is quite important. Beyond that, I have to say we also are leveraging a platform that's focused really more on analytical process development. Targeted LNPs, if you think about them, they're more complex drugs than the LNPs of nowadays, the approved drugs, if you look at them, and therefore need new tools to be able to elucidate, for example, the amount of coverage of a targeting antibody on their surface, whether you have unreacted species. These require new analytical process, analytical methods that needs to be developed and validated for the purpose of this. So this is a platform we leverage. Beyond that, you might have heard close to the end of last year, Cytiva was awarded an ARPA-H award for our ARPA Embody study, focused on in vivo, really developing LNPs for targeting solid tumors in vivo in the context of gene editing using CRISPR-Cas9 gene editing platforms. So this is another platform that we're developing, again, provide to our customers, but also to really lower the barriers of being able to use these modalities for these really complex diseases. And I also apologize, I'm going through these slides rather fast. I'd like to get us to the data, which is, to me, the more exciting part of this, which we've arrived at, so, and I think I still have enough time to share that. Reminder here, we are looking again at an in vivo targeted LNP in the context of targeted CAR T therapy. What that means is actually using the platforms that I've mentioned before. We are looking essentially for a LNP that is able to get into the spleen. Traditional LNPs, if you may, unoptimized LNPs, that you can see here as an example of which is LNP1, they traditionally prefer the liver if you inject them IV. If you administer them the IV, they will primarily go to the liver. That is well-known in industry. What we've done is leveraging those platforms, the ionizable lipids that we have, stabilizers, and really the compositional knowledge that we have, expertise, to develop other LNP compositions that have a bit more of a preference over the spleen. And please, if you look at this slide, kind of look at it, the half up top, where we're looking at biodistribution by IVIS in terms of these LNPs when injected intravenously. And we're seeing that there are several hits that even for LNP2 and 3, we're seeing a spleen to liver ratio of between 2 to 4 to 15, high points 34 here. That means even at 2 to 1 spleen to liver ratio, you're looking at 60, 70% of the LNPs being in the spleen over the liver. That's already a pretty good kind of starting ratio. But our goal is essentially to find an LNP that already from the start kind of goes there. So if you need to further help it get to the right type of cell, it has a better chance of being uptaken by that cell. If you look at the bottom here, what we're looking at is essentially, well, okay, we're heading with these compositions into the spleen. What types of cell are we hitting in the spleen with the unmodified LNP with this composition? So again, a novel composition, but without targeting moieties. What we're seeing here is we're actually hitting primarily macrophages and dendritic cells, so cells that traditionally would be the ones that tend to uptake LNPs, but in this context, the good part is that we are seeing some uptake in T cells. That is our target cells for this disease, so it's good, but I'd say it's a good start. Beyond that, the good part of this graph is also we're not hitting B cells, which is great because for this indication, if we hit B cells, that means we're actually helping them build up immunity to our CAR Ts. We do not want that, in fact. So not hitting B cells and hitting T cells is great. Just being mindful of the time here. So moving on with this composition. Another question we asked with our collaborator here. So the data from now on I'm sharing will be data that we've developed with our collaborator using our platforms. The question we've asked is honestly, is it actually, I mean, what we looked at the previous slide in biodistribution is expression. So are we not expressing in T cells because we're not getting into them, or is it potentially an activation challenge for these T cells? And that is the question that we tried to answer here by, our collaborators did, by co-dosing an CD3 adjuvant to essentially activate these T cells in vivo. And what we do find is that, in fact, the vanilla LNPs, again, unmodified, compositions with an adjuvant activating T cells, we actually do see expression of the encoded mCherry RNA reporter RNA construct in both the spleen and the blood and the lymph node. So actually, to a certain level, it could be an activation challenge also for these T cells. Like we are getting into them, in fact. More interestingly, I guess, we wanted to take this one step further and modify the surface of these LNPs with targeting ligands, in this case an anti-CD8, really antibody, really small VHH fragment. But the question is, when you're building out these targeted LNP therapeutics is, well, how many antibodies do you actually need on the surface? Like what do you need, like lower coverage, higher coverage? That's the answer we try to answer here on the left side with the blue graphs here. And indeed, if you increase the surface coverage of LNPs, of antibodies attached to LNPs here, as our collaborators have done, what they're showing is that, yeah, the fraction of these really LNPs that are getting into these cells increases. So higher coverage is better in this case. But I guess more importantly, if we're using a humanized mouse model engrafted with PBMCs, with a kind of human immune system, if you may, humanized mice, what we're seeing here and what we're showing on the right side is that with the targeting ligands, kind of the graph furthermost on the right, we are seeing that we're actually hitting about 80% of the T cells, CD8 positive T cells, both in the blood and the spleen. So it's not only the necessarily organ that we're looking at, these are also T cells that are circulating, which we want to be able to treat these type of diseases. And then actually, wow, surprisingly good on time. The last piece of this is looking at actual functional tests then. Armed with the knowledge that we have an LNP that is able to get into the right organs and hit the right cell types, be uptaken by the right cell types, what our collaborators have moved on to show is that if instead of a reporter, a fluorescent protein, they encapsulate RNA encoding for a CD19 CAR in this case, what they're seeing is that with the study that shows 3 doses of LNPs, following those 3 doses of LNPs in a pretty aggressive model of lymphoma in this case, we're seeing that even at the lowest dose study, which is frankly extremely low for CAR T therapies, 1.25 micrograms, they're already seeing a significant reduction in the tumor volumes of these mice. And again, with escalating dose, we're seeing a stronger and stronger reduction. Between the 3 mice in this study, you do see one that is a worse responder compared to the other 2. But in general, this showcases that really this kind of approach with the right design of a particle, again, that has a chance to get to your target of interest, target cell of interest, target organ of interest, modified further by a targeting ligand to get to the actual cells that are the effectors here, is a viable strategy that results in a really treatment that is able to again, to target these tumors and kill them in vivo after an in vivo administration. What we're following these studies up right now with the collaborators is higher order species. So there's ongoing NHP studies with this same, again, armed with this knowledge. This is where we are right now, and I'm excited to be, we're really excited to share that data once we're able to, probably in the second half of this year, or in a couple months at least, but I would say there are early indications of a good efficacy in NHP species also, for the same kind of design choice. And yeah, again, with 1 minute left, I'd like to just summarize. Our goal, again, is not to make our own therapeutics. We're here to leverage our platforms that we have to enable our customers' therapeutics and really their nanomedicine journey, your nanomedicine journey, if you may. So really happy to chat more. We have a booth. It's the greenest booth in the exhibit hall. I think 538, Erica, probably. But yeah, if you follow Cytiva green, you'll find it. That's the green that's on all our slides, I think. And yeah, I'd like to thank you all for joining us today, and happy to take questions. Maybe I'll skip this slide, but I'll say thank you because I see we're out. It's an anti-CD8 antibody. It's an anti-CD8 antibody, yeah. Yeah, so it's pretty classic in vivo CAR T approach, I would say, as a starting point. But it's quite flexible, I would say. Essentially, you have chemical moieties on these compositions, on these LNPs, and you can attach anything you want, really. On there, there is a bit of a question of making sure what you're attaching is pointing the right way. So analytically, it's a very interesting question, actually, for that purpose. But we have quite a bit of methods to analyze that. Thank you. Oh, sorry.
Great talk, thank you.
Thank you.
In general, do you see LNPs transfecting the tumors with a CAR T in a solid tumor environment? And is it a problem to make a tumor express a CAR?
That's yes, that's a really good, a really good question there. In general, probably not beneficial because you might start having a kind of building up immunity to the CARs you're transfecting. That being said, that's the idea is that if we design an LNP that really truly leans towards the organs of interest, have a better chance of getting to organs of interest, you do want to hit those T cells. Again, in this case, for example, by further modifying that surface. Again, but the key here is also to have a really good targeting ligand. Like the core of it is designing a right targeting ligand that's small enough that wouldn't change the properties of the particle too much, because if your base LNP is already decently good, you want to increase its efficacy, but not do something that would significantly change it.